Hi, I am Tzalik Naani from Carmel Diagnostics. I am pleased to be with you and present to you our Fertissimo system for better embryo selection in IVF process. The system substantially improves the success rate in IVF, which is very important for this system. Uh, in the coming video, you are going to see three parts. One of them will be 2 minutes pitching. After that, there will be a video that shows you the operation of the system with all the details, and we'll finish with a PowerPoint presentation where I'll explain the system, I'll describe the company, and I'll tell you what we are looking for, and I'll show our achievements. So please enjoy and have a good time. Bye. One of 6 or 7 couples in the fertility age suffer for infertility. For each one of them, this is the whole world, and they will do everything in order to have a baby. Can you imagine a solution that will help these people bring a baby home? There is a solution, IVF, in vitro fertilization, which brought millions of babies so far. However, the success rate is very poor. Here we come. I am Tzalik Naani, the CEO of Carmel Diagnostics, a company specialized in the IVF sector. We have developed and patented a system that substantially improves the success rate of the IVF process, saving money to the healthcare system, shorten the time, pain and cost to achieve pregnancy, and help the couples bringing a baby. By providing clinics with efficient embryo selection using oxidative stress as a biomarker. This non-invasive technology is innovative and disruptive in comparison to the state-of-the-art morphology. We conducted successful clinical studies testing hundreds of embryos, and the results have been published in leading magazines and in professional trade shows. We have an addressable market of $500 million through our pay-per-test business model. We have a dedicated, experienced team, and among our advisory board, there is a Nobel laureate. Our goal is to become the new standard of embryo viability assessment and get 10% of the world market in the 5th year of sales. Thus, we are looking for an investment of $4 million with an ROI of 3 years. We invite you to talk to us and meet us to have more details of our plans and explore this great opportunity.
Fertissimo analysis of an embryo, step-by-step operation. 15 microliters of embryo culture media is drawn from the dish using a pipette and inserted into the cuvette. In Station 1, sample shaking to obtain a unified layer. In Station 2, sample drying using a vacuum. Station 3, sample heating and scanning. Emitted photons are collected. Scores of the embryos in descending order are displayed on the device's display.
A disposable tray containing 8 cuvettes to test embryos of the same patient. When needed, additional trays are used. 15 microliter of spent culture media from each individual embryo are inserted into the cuvette using a pipette. The samples are spread over the cuvette to get a unified layer. The operator writes the embryo number in a designated place in the tray. This process repeats for all the tested embryos of a patient. When ready, the operator brings the tray to the system. The operator logs in, selects job title and her name and presses login. test detail screen is open. The operator types in the patient ID. Default value of embryo day and medium type appear. The operator may modify them if needed. The drawer is opened. The tray is inserted and locked. The operator assigns embryo numbers to all the relevant cuvettes. This process is done twice, and the entered data must match in the 2 rounds. Vacuum drying and scanning processes start. The system displays which cuvette is in which operation, as well as showing the progress. After scanning all the cuvettes, a sorted list by embryo score will appear to lead the embryologist in embryo selection. The selected embryo is transferred to the patient's uterus, and pregnancy will be hopefully achieved.
Hello, thank you for the opportunity to present to you our system and our product. Our product comes to improve substantially the results in the IVF process. The problem, as we know, is a big one and attacks a big percentage of the population. There is a solution, IVF, in vitro fertilization, but the success rate is very poor. The answer is better embryo selection. Embryo selection is actually the holy grail of the IVF. The challenge is to find the embryo with the best, highest probability to conceive and achieve pregnancy. We built a system that improves the results by something like 20%, showed in clinical trials. It takes about 8 minutes from start to end per embryo. We use oxidative stress as a biomarker, and we use also artificial intelligence, bioinformatics methods, and machine learning in order to improve continuously, using feedback from the field, the correlation formula of scoring the embryos. Few words about the company. We spent about $7 million developing and validating the system. We had clinical trials showing good results. We formed solid partnership with IVI Valencia, the biggest and best IVF clinics network in the world, and we got endorsement from the industry. We plan to establish presence in China, to have manufacturing here, to distribute the system with cooperation with local companies, to go on with R&D in cooperation with local industry and academy. And in order to do that, we need an investment of something like $4 million that will bring us to the market with an excellent product. Thank you very much.